Hello class, welcome to the second lecture for chapter 3, Nucleotides, Nucleic Acids, and Genetic Information. So this is the second part of the chapter, and in this part we're going to discuss nucleic acid techniques. These techniques are going to include polymerase chain reaction, or PCR, sequencing, restriction enzymes, gel electrophoresis, and a little bit about genetic engineering. So let's start with polymerase chain reaction, or PCR. Of using a PCR is that you can make many copies out of a DNA molecule. So if you have a small amount of DNA and you want to make a lot of it so you can then use it for something else in the future, you're going to put it through PCR. And this process of making many copies of a DNA molecule is also called amplifying. So in the first cycle of PCR, we start with our original target, which is made of duplex DNA. That means it has these two strands. These strands are then separated by heating. And these strands are acting as the template. Another word for the separation of the strands is denaturing. So the first part of any PCR cycle is the denaturing step. Then after that, the temperature is cooled slightly and primers are then annealed onto the DNA template. There's one primer that goes in each direction. Primers are short DNA sequences that are going to bind to either end of the region of DNA that we want to make a copy of. So they are sort of like the bookends of your PCR. Once your primers have annealed, which we show here, we then have to add DNTPs, or deoxynucleoside triphosphates, as well as a DNA polymerase enzyme. This enzyme has to be thermostable because, like we just mentioned, the first step of this reaction is heating it up. So we want our DNA polymerase not to be damaged by heat. Most enzymes that we see are optimized to work at our body temperature if they're in our body, which is about 37 degrees. For the PCR reaction, the DNA polymerase that we use is from a bacteria actually called Thermus aquaticus, which we abbreviate TAC, so we call it TAC polymerase. As you might guess from the name Thermus, this bacteria is really stable at high temperatures, and so its DNA polymerase is also really stable at high temperatures, so it can survive our PCR reaction. DNA polymerase can only add on to the 3' end of an existing DNA strand. It cannot begin a new one, and this is why we attached our primers first. So once we have our primers on, our DNA polymerase can try to extend from the 3' end of each of our primers so that they can eventually meet on the other side. But to do this, DNA polymerase needs another ingredient, which are deoxynucleoside triphosphates, and you add all 4 of them. So you have ATP, TTP, CTP, and GTP. So these, as you may be familiar with, are the 4 nitrogenous bases that you find in DNA: adenine or adenosine triphosphate, thymine or thymosin triphosphate, etc. So once you have the building blocks and you have the DNA polymerase, you can then the DNA polymerase will then extend along the primers as long as you allow them to, as long as you give them time to. And that creates what we call these variable length strands, because these strands can extend some length out in either direction, and the only thing that affects it is basically the temperature and the time that you allow it. So once you've created these variable length strands, then it's time to enter cycle 2. So once again, we start the cycle by denaturing our DNA template. We heat it up to separate the 2 strands, and we add in our primers. The primers are allowed to then anneal onto either side of our template, and that is the first part of cycle 2. Once we've done that, we then keep adding on our DNA polymerase and our DNTPs, which are in our reaction mixture. We'll then go ahead and extend the template. So this time, if you're working off of the original DNA template, it's very long, right? It contains the whole genome. But now you've also, in your first cycle, you've created 2 new strands based off your primers, and these strands are going to end at either end of your primer, right? So now, as you continue to go through PCR cycles, you're going to get mostly copies of those strands that have a definite endpoint on either side that began from the primers, and then you'll create what are known as unit length strands, which are essentially the strands that are the length of the DNA molecule that is in between your forward primer and your reverse primer. And then as you continue to repeat the cycle, you'll make just thousands and thousands of copies. So the number of copies that you create can actually be calculated based on how many cycles that you run of PCR. So the number of copies you make is the number of original DNA strands multiplied by 2 to the nth power, where n is the number of cycles that you run. So as you can see, this adds up really, really quickly because this is an exponential equation. So why did we talk about PCR? Well, PCR itself is pretty cool. It's a great way to amplify DNA if you want to then use that DNA to do something else with it, or just look at the DNA itself. But what else can we do with PCR? So one common variation on PCR that you may have heard of is called reverse transcription PCR, or RT-PCR. For this one, you actually start with an RNA template. And then the first step of this reaction is using a reverse transcriptase enzyme that uses RNA as a template to make DNA, complementary DNA, also known as cDNA. And this can be really important when you're looking at your genes that are expressed, as opposed to just the way that genes are encoded in your DNA genome. So as we've discussed, DNA gets made into RNA, which gets made into protein. So if you're interested in which RNAs are getting actually made into proteins, this reverse transcriptase PCR is a great way to look at RNA from, for example, gene expression. It's also a great way to look at other types of organisms that use RNA for their genome. For example, there are a lot of common viruses which do this. One virus which you've probably heard of is SARS-CoV-2. It has an RNA genome. So if you want to look at its genome, you have to always start with reverse transcription to create complementary DNA, which we can then use our standard techniques to work with. So once you've made your cDNA, it can then act as a template for all the remaining PCR cycles, just like we talked about a couple slides ago. Another type of PCR is known as real-time or quantitative PCR. This is a little bit confusing because real-time and reverse transcriptase both have the initials RT. So if you just refer to RT-PCR, it may be a little bit ambiguous, so it's helpful to call the second one quantitative PCR or qPCR. So qPCR actually, instead of just making a lot of copies of the DNA, it actually keeps track of essentially how many copies you've made. So you do this by adding a fluorescent probe with your reaction. And fluorescent signals are extremely sensitive because fluorescence is so bright. So when you've gathered enough copies of your DNA, the probes will fluoresce strongly enough that the machine detects them. And based on how many cycles of PCR it took for you to see that fluorescent signal, you can back calculate to figure out how much DNA you started with. So why is this interesting to us? There is a clinical connection here where real-time reverse transcription PCR can be used as a diagnostic test. So, for example, with SARS-CoV-2 again, we have that RNA genome that I just mentioned. So you're going to first use reverse transcriptase to make it into complementary cDNA, and then you can use primers and probes made out of DNA for a quantitative PCR reaction. And this can tell you how many copies of the virus you have within your body. If you have many copies, that indicates you may be in the middle of an infection. If you have very few copies, this might indicate that you're towards the end of an infection, or it could even just be human error. Maybe there's a little bit of genome in the air that you can ignore. Another helpful use for this, like I mentioned, is to look for host RNA or any organism that uses DNA to make RNA to make protein. You can look at how much RNA you're making for a particular gene, and that can tell you essentially whether you're making a lot of that gene and making a lot of protein out of that gene to then do something inside your cells. So now that we've talked about PCR or making copies of DNA, Something similarly related is sequencing DNA. So it might not be enough just to have a whole lot of copies of a DNA molecule. You might want to know the actual sequence of nitrogenous bases in that molecule. So the sort of original method is called the chain terminator method, or the Sanger method, which is illustrated here. So you start just like PCR with your template DNA. Your one strand is shown. So you have your nitrogenous bases shown at the bottom, and they're connected with a sugar and a phosphate. This is the 5' end, and this is the 3' end. So just like with PCR, you also start with the primer, and you add these DNTPs, and you use DNA polymerase. And this sort of demonstrates how the general scheme of what you're looking at. So you're looking at what happens when you add one base at a time. So it's called the chain terminator method, because the end goal of this type of method is to create a number of chains that each have a differing length, and then you can sort of put them in order, one base at a time. So here's a more description of how that is achieved using this method. So at the top on the left, we have our normal deoxynucleoside triphosphate. This is the type of nitrogenous base using A, T, C, or G that we would use list like in any PCR reaction. It has a 3' OH and a 5' phosphate, and this is the building block that we need to build up our normal DNA molecule, and you attach new molecules going this way, right? You have the 5' direction towards the 3' direction. So on this 3' OH, you would need to add on your next DNTP. So in order to terminate the chain, so you can't build any more, you actually use what's called a dideoxynucleoside triphosphate, or a DDNTP. So here, not only do we have the 2' H, which indicates it's deoxy, it's for DNA, not for RNA, for our deoxyribose. We also have a second deoxy at the 3' position. And this means we can't add on any further base pairs after this 3', and because you need that oxygen there in order to add on. So this means that this base pair that you would just add it on is going to be the last base. So this is sort of illustrated here on the right. If you start with a primer that's complementary to this template that's shown on top, the primer will try to extend in the 5' to 3' direction, and you have all 4 of these dideoxynucleoside phosphates present in your mixture, as well as the normal deoxy ones. The dideoxynucleotide ones are labeled with dyes, so each one is a different color, and so you can end up having different, depending on exactly where your chain terminated with which one of these, you will end up with different segments of DNA that all differ in length by exactly one base, and each base will give you a different color. So based on the length and the color, you can figure out the exact sequence that this DNA polymerase used to fill in from the template. So another method for doing this is to carry out the reaction in 4 different containers. Each one of these containers has a different dideoxynucleoside triphosphate, as well as the 4 regular deoxynucleo ones. And it's the same idea. So we have 4 different colors. Each one terminates the chain at a different position along the template. And then we can separate on a gel, so we can use a sequencing machine to read the color. And we can also separate each of these bands on a gel to look at the length of the strand, as well as the terminating dideoxynucleoside. So for example, our shortest strand is going to go all the way to the bottom of the gel. And this will be the 5' end of our copy, because that's the earliest part. So when we're copying this DNA template, we start at the 5' end with our primer, or with our match, right? And we know that our match to A is T. And we know that it's T because it only happened in this pink tube that had dideoxythymosine triphosphates in it. So, thymidine, excuse me. So then as we go along in increasing chain length, we can see, based on the colors from each of our 4 reaction tubes, we have a T, then we have a C, which is complementary to this G, then we have the G, which is complementary to the C, etc., as we go in longer and longer lengths along this gel. And we'll talk a little bit more about gel electrophoresis on upcoming slides. So, another type of DNA sequencing, oh, and I just wanted to mention that this type of chain terminator method is really great. It's very accurate, but it's only really useful on fairly short lengths of DNA. If you wanted to sequence a very, very long piece of DNA with thousands and thousands of bases, this is just an inefficient method that becomes more difficult the longer your DNA template is, because you have to count on making all of those in between strand lengths, and the more DNA you have in length, the more challenging that is to make sure you get every single one. So there are what we call next-gen sequencing techniques that we can use to sequence lengths of DNA that are as large as an entire genome. And one type of this type of sequencing is called pyrosequencing. And there's a link to a video here in the slides, and then I'll explain a little bit on the next few slides here. So for pyrosequencing, we start similar but different. We have a forward and a reverse primer, but one of the primers is biotinylated, so it has a biotin molecule, its protein attached, and that helps it to stick onto a surface. And then, sort of like we saw with PCR, we add DNTPs to the reaction mixture, but we only add one of them at a time. So first maybe we'll add ATP, and then we'll wash everything away, then we'll add GTP, and we'll wash everything away, etc., etc. If the DNTP that you added is complementary to the next base pair in the template, then the DNA polymerase will allow the incorporation of this DNTP. So in this case, when we added TTP, the T was able to hydrogen bond to the A, and the polymerase was able to add it onto the growing DNA chain from 5' to 3'. And every time that one of these nucleosides is incorporated into the DNA strand by a DNA polymerase, the reaction always releases an inorganic pyrophosphate, or a PPI, which is shown on the bottom here. And that's part of why this is called pyrosequencing, because we're looking at the release of a pyrophosphate. So each pyrophosphate released by this reaction, that is catalyzed by the DNA polymerase, is converted into a TP by a sulfurylase enzyme, which is shown up here. And ATP then fuels the conversion of luciferin to oxyluciferin in this enzyme called luciferase. And this luciferase reaction is accompanied by the release of light. So every time you see a flash of light, you know that you just incorporated a DNTP into your growing DNA chain. And that light is recorded by a machine that's part of this sequencing apparatus. And after that, another enzyme called apyrase then is going to degrade all the unused DNTPs and ATPs that may have been created in that last cycle, and then you're ready for the next cycle where you add another DNTP. And you continue this, until you reach the end of your chain. So each successful incorporation of a nucleotide, like I mentioned, results in a burst of light. And the area underneath the curve is proportional to the number of nucleotides that were just added onto the growing DNA strand. So you can then read the DNA sequence of the complementary strand in a 5' to 3' direction. And this allows you to then determine the sequence of the template strand. So here, we added 1G, 1C, then a T was flowed through that did not get incorporated because we do not see a flash of light. Then the A was incorporated. And here with the G and the C, you see that the G and the C, the area under those curves is about twice as much as we saw for the other ones. This means you incorporated 2 of those DNTPs at each of those positions. And finally a T. So just to make this really clear, let's talk about what exactly happened based on this light diagram that we're looking at. So our complementary sequence from the 5' end has a G, a C, a T, Sorry, it doesn't have a T. Excuse me. Can't read my own handwriting. Excuse me. So we have a G, a C, an A. Then we have two G's. We have two C's. And then we have a T. And then that is at the 3' end. So our template sequence must be exactly reverse complementary to the template sequence. Sorry, the template is a reverse complement to the complementary sequence that we read out on this light diagram. So our template sequence, therefore, goes in the reverse direction, right? So it's anti-parallel. So the 3' end of the complementary sequence is the 5' end of the template sequence. And then we simply have to determine what the complementary base pairs are. So T, A, C gives us G, G, C, C, T, G, C, and then that ends at the 3' end. So that now we know our original template DNA had this particular sequence. Often we like to write our DNA sequences from 5' to 3'. So if you wanted, you could also go ahead and turn this around and just know that our template sequence from 5' to 3' actually reads A, G, G, C, C, T, G, C. It doesn't matter particularly which way you write them. It's common to write it from 5 to 3, but what's the most important is that you label which end is your 5' end and your 3' end, because that makes a really big difference, obviously, in knowing what the actual order is. And there we are. So another type of next-gen sequencing is called Illumina sequencing. And again, this helps us to figure out the sequences of very long DNA strands. And so the next few slides here have pictures from Illumina. So one example of how we might use this is to actually sequence the full genome of SARS-CoV-2. So before, when we were talking about RT-PCR and qPCR, we were looking at amplifying specific genes that we're already aware of, that we already can make primers to, so we already know the sequence of those genes if we're doing qPCR. Here with Illumina, we don't know the genome sequence at all, so we start sort of from something generic, and that way we can read out the very specific sequence so that we don't know anything about. So as an example, SARS-CoV-2, as we know, has a single-stranded RNA genome. To do anything with that, we have to make it into DNA. So we use reverse transcriptase, just like we talked about before, to make that cDNA. The cDNA, we then sequence it using this high throughput technique, such as Illumina, which we'll describe in the next few slides. And then based on that, we know that our complementary DNA is complementary to the original RNA sequence, and that can help us figure out the full genome. So the way we do this in a little bit more detail is first we make a library. So we have our genomic DNA, or in the case of SARS-CoV-2, we would have cDNA. This genomic DNA is then fragmented into smaller pieces, and little adapters are attached to each end of the fragments. And this creates what's known as a sequencing library. So these special adapters are then able to actually stick onto the surface of this flow cell. And so you have different areas of the flow cell with those fragments attached that have those adapters on the end. And then we also have lots of other adapters on the surface of the flow cell. And this allows for what we call bridge amplification, where each little fragment of DNA then gets copied by little mini PCR reactions and stuck onto the surface into what are known as clusters. So now those 4 fragments have been made into 4 clusters. And then we're able to go ahead and sequence them. And the way we do that is sort of similar to what we talked about with Sanger sequencing, where each fragment is essentially a different length, and each one terminates with a different nucleotide, right? So we have A, T, C, and G, and each one of them is represented by a different color. And when we look at the flow cell, we see each of our clusters that goes in a different color. So as we go through the PCR reaction, just like we talked about with pyrosequencing, we're adding one DNTP at a time. And so after we added that particular DNTP, if we see a color, we know that that was the next nucleotide that was added on. So in the case of this cluster that we're following, that's circled with a white circle, First we added on a T, which is green. Then next we added on a G, which is blue. Then we added on a C, etc. So we know that after these 4 cycles where we added these 6 particular cycles where we added on those particular DNTPs, we were able to read out this particular cluster has this particular sequence. So, and so just to be clear, we're using the cluster to add on new DNTPs that are complementary to the cluster. So we're essentially adding on one DNTP at a time to help us see, based on the color, which DNTP has been added. So once we're done copying all the DNA and creating all these little complementary fragments, they can then get aligned. And you can also use a reference genome, and this essentially helps us to put together all the different pieces in the right order, because you have all these little chunks that we created at the beginning, and they have to all be aligned together. It's almost like you just shredded a big document, and now you want to put back together the page correctly using just the shreds. So that brings us to some other DNA techniques that go beyond amplification and sequencing. So we're going to talk a little bit about genetic engineering. So what is genetic engineering? Genetic engineering we're going to define here as the process of modifying DNA using molecular biology techniques. And so this can include things like inserting DNA from one organism into another organism. So for example, we might want to make a pathogenic virus gene inside of a bacteria or some other organism that can help us make that gene, but without having to use the actual dangerous organism. And so this is known as creating a transgenic organism when you have one organism that has a gene from a different type of organism in it. We also might want to use DNA modification through genetic engineering to make point mutations on a DNA sequence. And this can help us determine if we then try to make that DNA into RNA and into a protein, if we make mutations at various points along DNA, we might affect the function of that protein that we eventually make out of that DNA. So making point mutations can help us figure out maybe what is the active site of a protein. That might give someone a cell its function, which could also lead to a disease phenotype. So what are some applications of this? I already mentioned a few. So this might help us study and develop treatments for many diseases. If we can determine which part of the protein is functional, we might be able to help find cures for that disease. It also can help us to just create new DNA sequences and new proteins that could have applications in, for example, treating disease. And we can also use these types of techniques to express large quantities of a protein using bacteria or yeast as our little machine that makes a lot of protein for us, which then we can use to study whatever we'd like about the function of that protein, etc. So what are some challenges of genetic engineering? The first challenge we should always consider is ethics. So it's very complicated to think about what are we doing when we make changes to a human gene or a human protein? Do we want to make those changes within humans? What does that sort of mean for the morals of whether that's okay or not to do? A lot of people have different opinions on this. It's something that's great to talk about. Other challenges we have that are more practical are introns. So you may know that most eukaryotic DNA, the genes have both exons and introns, where the exons code for protein and the introns are spliced out by splicing machinery. So if you try to make a eukaryotic gene directly by a prokaryotic organism, for example, a bacteria, it won't be able to chop out those introns for you correctly. So you will need to keep that in mind when you're amplifying your gene and perhaps remove the introns yourself before you ask the bacteria to express that gene for you. Other challenges that you might face in the lab are post-translational modifications. So we've talked about the function of the ER and the Golgi. When you're making proteins, they can add on little bits and pieces, for example, sugars, onto proteins. And this is different in different organisms and different, and in particularly between eukaryotes and prokaryotes. So if you have a lot of proteins that you make won't function correctly unless they have the correct post-translational modification. For example, if a lipid is attached to them, that helps keep it attached to a cell membrane, and suddenly you didn't put that lipid on, the protein can't get attached to the cell membrane anymore, and it might not be able to do its job because it can't find its home. So these are the sorts of things we need to think about when we're trying to use one organism to express DNA that originated in a different organism. So what are some practical uses of genetic engineering? We talked about how it's great to make recombinant protein using different organisms to help you out with proteins that you want. So some really important applications of this, one is making human insulin for the treatment of diabetes. It would obviously be endlessly complicated to take insulin from just other humans, so why not use a recombinant expression system to make human insulin, but perhaps asking a bacteria or a yeast to do it for you. Similarly, for treatment of other diseases, we have a list of a few other recombinant proteins here. It can also be used for vaccination. So here we have the example of the hep B surface antigen, which is made recombinantly and then used in the vaccine to help prevent hep B in people who are exposed. And it even has more sort of mundane uses. So we can use recombinant protein expression to make vegetarian rennet, to make cheese, for those who don't want to get rennet the traditional way, which, as some of you may know, rennet is an enzyme that cows use to help break down milk, and it's also used in making cheese. So the sort of original way to source rennet was to actually take it from the stomachs of cows, of baby cows, calves. So it's sort of a much less cruel way if we can just make our own recombinant rennet without having to sacrifice those calves, and we can still enjoy some delicious cheese. So how do we go about doing genetic engineering? We want to take DNA from one organism and put it into another, but we don't want to take the whole genome, right? We want to take maybe one gene or even one piece of a gene that we're interested in. So how do we do that? We start with cutting out the gene that we want. And the way that we really commonly do that is use restriction endonucleases, which colloquially a lot of people just call them restriction enzymes. And these restriction endonucleases can cut DNA at very specific sequences. Their actual original purpose, we kind of borrowed them from bacteria, but what bacteria actually use them for is part of their own immune system. So they use it to look for foreign DNA. For example, if a bacteria was infected by a virus, bacteria then make these restriction endonucleases that will go look for specific sequences that are maybe only found in pathogen DNA, and chop up that DNA so that the bacteria's virus can't replicate inside the bacteria. And so anyway, we can now borrow those restriction endonucleases for our own purposes in the lab. So that's a long way to say that restriction enzymes are named, often for the species that we borrowed them from. So these two that we are showing on this slide, they originated in E. coli. That's based on the first three letters, E for the genus and CO for the species coli. And then the R1 and the R5 refer to different strains that they might have been isolated from, or just how many restriction enzymes were isolated from that particular species. So, like I said, each one of them cuts a DNA at a specific recognition sequence. So EcoR1, which we're showing at the left, looks for the sequence GAATTC. And something to note, actually, is that all of these recognition sequences for restriction enzymes are actually palindromes. So they both, from 5' to 3' will read GAATTC. The complementary strand also reads GAATTC from 5' to 3'. And this EcoR1 is actually going to cut right after the G in both circumstances, just like these arrows show here. And so once it cuts in between here, you end up with a sort of hanging end that, once these strands are separated, is what we refer to as a sticky end. Because each of these bases, now that it's not hydrogen bonded with its complementary base, once you separate these strands out and cut them up, they're going to be sticky, right? They're going to be looking for a complementary strand of DNA so that they can hydrogen bond to them and be happy and stable. A restriction enzyme such as EcoR5, which we show on the right, has a very similar recognition sequence. It's also a palindrome, GAT, ATC, GAT, ATC, except EcoR5 actually cuts right down the center of its recognition sequence. And this is what we refer to as a blunt end. Blunt ends are a little bit less sticky than sticky ends, right? Because they don't have just bits of one strand hanging out looking to hydrogen bond with a complementary strand, but we can still stick them together with other blunt end pieces of DNA. So here is a list of some restriction enzymes and their recognition and cleavage sites. So the recognition sequences, often in shorthand, we'll just write one strand of it from 5' to 3', since, as we know, they're always complementary to each other. And then these arrows shown in the diagram show where the cutting would be. So we've taken as an example from this chart the TAC1 restriction enzyme, which has a recognition sequence of TCGA. Oops. So as an example, we can look at the complementary sequence. So if we want to fill that out to sort of get that picture of the full double-stranded DNA molecule that it would be cutting, we can simply write the complementary bases. So T is paired with A, C is paired with G, G is paired with C, and A is paired with T. And again, we're going to cut between the T and the C on both strands. So you can think of making just a line, and then in between here are where you'd see the sticky ends forming. So now that we've looked at the restriction enzyme TAC1, and we have its recognition sequence, which I've written again here, now we can look at a longer strand of DNA and figure out where exactly TAC1 might cut that sequence of DNA. So we can see our recognition sequence right here in the middle, TCGA, TCGA, and we know that we're going to cut between the T and the C. So we can draw these little cuts here, and then we can sort of even attach in the middle where the sticky ends are going to be. So the sticky ends for each one are going to be a G followed by a C if you're going from 3' to 5'. So these are, after we cut this molecule, this is what it would look like. So this top part here only has 4 bases. This bottom part is going to have all 6 of these bases, and this G and the C are the sticky end. So if we use another restriction enzyme that produces the same sticky ends that also contains a G and a C, then if you mix together all those sticky ends in a beaker or in a cup or whatever, any of those sticky ends can then find each other and stick to each other, form hydrogen bonds. And so in this example, we used TAC1 to cut the sequence at the top, and then on the bottom we used a different restriction enzyme that has a different recognition sequence, but that creates the same sticky ends. So here, if our TAC1 enzyme cuts here, our sticky ends are C and G. Our HPA2 MSP1 enzyme cuts, in this case, after the C, but we still created the same sticky ends, which are C and G, right? So after we cut, we have these matching sticky ends, and that allows any of these strands of DNA to then stick back together. These are then sealed with an enzyme called DNA ligase that actually creates the sugar phosphate bond in the backbone, because all the sticky ends are going to do is stick together with hydrogen bonds in between the nitrogenous base pairs. So why did we go through restriction enzymes for so long? Well, because it's a really common method that we use when we want to insert foreign DNA into an organism. So depending on the length of the gene, that piece of DNA that we want to insert, we can use a different, what's called a vector. So all 3 of these are called vectors. So we have plasmids, which can accept DNA sequences up to 10 kilobases, which is maybe 1 or 2 genes, not a huge amount. Then we have bacteriophage lambda. Bacteriophage is actually a virus that infects a bacteria, and we can insert foreign DNA into there up to 16 kilobases. And finally, we have artificial chromosomes, which can use up to thousands of kilobases. So on the right here, we have a picture of an enzyme. And our enzyme has a couple of really important features. I'm sorry, I said enzyme. This plasmid that we show here on the right has several really important features that, that help us know whether or not we incorporated our gene, because we had to come up with sort of clever techniques to figure out, is what I think I'm doing in the lab really what's actually happening to what I'm working on? So when we want to insert this foreign DNA, we look for several important features. So, but first, how do we stick it in? We, just like we talked about, we have our foreign DNA, we have our cloning vector. So, for example, our plasmid. And then we have to cut our foreign DNA. We usually create sticky ends, and then we'll cut our plasmid with sticky ends that are complementary to the sticky ends we created on our foreign DNA. That allows the sticky ends and the foreign DNA to anneal together, meaning the hydrogen bonds are created between the base pairs. And then we use DNA ligase to then covalently join the sugar phosphate backbone of those DNA strands. And that creates what we call a chimeric DNA, which has some foreign DNA that's inside the larger vector. We can then use that vector to insert into another organism. So now we can get on to some features of plasmids that help us use them as the tools that we'd like them to be in terms of genetic engineering. So first, every plasmid has an origin of replication. The origin of replication is basically directions for the organism that you're sticking the plasmid into that tell it to make sure to replicate this particular plasmid. Different plasmids might have different origins of replication that might help the bacteria make fewer or more copies of those plasmids. And so it's important to have a plasmid that you know will make the appropriate number of copies that you want. The other important thing we have are restriction enzyme recognition sequences. So just like we just talked about on the last slide, we had to create those sticky ends in our foreign DNA to stick them into our plasmid. So our plasmid has a number of different restriction enzyme sites that are shown all around the outside here, for example. I didn't circle all of them, but you get the idea. And so some of those, at least one of those sets of recognition sequences will hopefully be compatible with the foreign DNA that you want to insert into your plasmid. And another really important feature is this ampicillin resistance gene, which you see in green on the left side. So ampicillin, this gene allows the plasmid to, it has directions for making an ampicillin resistance enzyme, and that allows the whole bacteria to be resistant to ampicillin. So now you'll know that if the bacteria itself is naturally not resistant, and you add this plasmid in, then you know that only the bacteria that took up your plasmid will survive if you grow them in an environment that contains this antibiotic. And so that way you can ensure that going forward you only pick out bacteria to keep propagating that contain the plasmid that you put in them. All the other bacteria that didn't take up plasmid you can discard because they're not going to be helpful to help make your gene of interest. The other really important feature of a plasmid is the lac operon. So our AMP resistance gene is going to help us select for bacteria that took up our plasmid. The lac operon is actually going to help us select for bacteria that took up plasmid that contains our gene of interest. Because, and as you might have learned about lac operon, hopefully in like biology class, lac operons have inducible expression. So let's actually go on to the next slide to explain a little bit how we use this to our advantage. So sorry, that's going to come up in a couple of different slides. In a couple of slides from now, I'm so sorry, I can't speak today. So first, let's talk a little bit about once you've made your plasmid, how do we look at it? How do we visualize DNA or protein? So we do this by a process called gel electrophoresis, and this helps us to separate molecules by size, shape, and charge. So when we're talking about DNA, we use a gel that's made of agarose, which is a type of sugar that can cross-link, and different lengths of pieces of DNA will travel through that agarose gel at different speed, pretty much depending on their size. So small pieces of DNA will travel further, faster than large pieces of DNA, which get sort of stuck in that gel matrix and they can't move as fast. Once we've allowed the DNA to travel through the gel, we then stain it with a dye called ethidium bromide, which is shown over here. This ethidium bromide intercalates itself in between the DNA strands, so it basically breaks up where the nitrogenous bases form hydrogen bonds with each other. And it glows under UV light, so it allows you to visualize where the DNA traveled to in your gel. We can also, similar but different, use gel electrophoresis to visualize any proteins that we might want to look at. We use a slightly different type of gel that's made from polyacrylamide, and we can use a different stain that's called Kumasi blue that prefers to stick to proteins, as opposed to ethidium bromide, which prefers to stick to DNA. And a protein gel is actually shown on the right in this diagram. Protein gels tend to be run vertically, so you have a thin layer of gel in between two pieces of glass, and you have these wells at the top where you add your sample, and based on charge and size, the proteins run down the gel, and again, the smaller proteins go faster, and they get to the bottom, and the larger proteins get stuck near the top. And once the protein has run through the gel, then you add that Kumasi blue dye to visualize all the proteins that are inside your gel. So the concepts for both of these types of gels, big molecules move smaller distances. Small molecules move bigger distances. And so to get a picture of that DNA gel with that ethidium bromide, so first of all, DNA gel on the left that we see here, we run it horizontally. We lay the gel flat and we add the DNA, but same principle, we use charge to ask the DNA to move down the gel. And then to visualize the DNA bands, we have ethidium bromide, which we see on the right. And ethidium bromide glows that sort of bright orangey pink color against the darker background of the gel. So that brings us back to our plasmid. Once we add our gene into that plasmid, how do we know that, one, that our gene got put into the plasmid, and two, that the bacteria that we want to grow our plasmid actually took up the plasmid with the gene inside of it? So here is where our lac operon comes in. So our lac operon has the LAC-Z gene in it, and that gene encodes an enzyme called beta-galactosidase, which we will call beta-gal. Beta-gal's normal job is to cleave lactose and help the bacteria digest lactose. But in this case, we add a compound called X-gal, which we show here on the right, which is also cleavable by the beta-gal enzyme. But when we cleave X-gal using beta-gal, we end up with a blue product. So we can use this feature to know whether or not our beta-gal is working. If beta-gal works, we'll get a blue product. If beta-gal doesn't work, we won't get any blue color. So how do we use this to our advantage in our system with our plasmid? That's where we insert our gene. So this polylinker region has a number of different restriction enzyme recognition sites, and that is where we most often want to stick our gene of interest, because if we do that, we're going to interrupt this whole purple LAC-Z gene. So if our gene was correctly inserted into the plasmid, the beta-gal enzyme doesn't work anymore because we interrupted it, its code. But we still have our ampicillin resistance gene to show that to allow our bacteria to survive in antibiotic. So bringing us back to our blue color, if we're able to insert our gene into that polylinker region, we don't make beta-gal, and our cells are colorless. So here we have a plate. We can have our plate have our ampicillin antibiotic, so that way only bacteria that took up our plasmid are able to grow on this plate. And in addition to that, we can see both colonies that are blue and colonies that are white. Colonies that are blue have a functioning beta-gal, so that means the plasmid does not have our gene of interest in it. On the other hand, our white bacterial colonies don't have a working beta-gal because they can't cleave X-gal to make that blue color. And that means that we successfully interrupted beta-gal by adding our gene of interest. So if we want to go forward using our recombinant gene that we made, we want to pick out these colorless colonies. So that brings us to our last method of genetic modification, and this is called, and we're going to call this site-directed mutagenesis. Site-directed mutagenesis is a really great way to introduce small DNA mutations into a gene to then see what effect that might have on the RNA or the protein going forward. That might, for example, help you study a disease state. So one simple way to do this is using PCR, which is illustrated here on this slide. So in normal PCR, which we've talked about before, we have this synthetic primer, which is exactly complementary to our gene. If we want to do site-directed mutagenesis by changing just a couple of base pairs, we create a slightly different primer, right? So our primer actually isn't an exact copy, right, of our template. It has a few mismatches. So for example, here our primer has a C, our template has a T. Those are not going to hydrogen bond with each other. And over here we have a G and a T in our primer. G does not match with A, T does not match with C. So again, those parts of our primer don't match our template. But there's enough of the rest of the primer that the primer can still anneal to that gene of interest, and we can still use DNA polymerase to extend that DNA fragment and copy the rest of the gene. So a few mismatches now, when we amplify this new strand that we just created, those mismatches are what's going to then be amplified by further cycles of PCR, which we show here. So our mismatched primer, we add our DNTPs, just like we would for normal PCR, and now we created an altered gene that fits onto our template. And then when we denature these again and make copies of them again, eventually our altered genes are going to vastly outnumber our unaltered genes. So with that, I hope you understand a little bit more about these nucleic acid techniques, and you are ready to go ahead and answer the practice questions document.